Proteins are microscopic biological machines that run our bodies. They have a wide variety of functions. They help our muscles contract, transport oxygen to cells, and help us convert food to energy our cells can use. We know that amino acid sequences and their interactions with one another determines the protein's final structure. But how exactly do proteins go from this to this? Levanthal's paradox states that it would take longer than the age of the universe to sort through all the combinations of a protein structure to find the right one. Yet proteins can do this in milliseconds. So how can they fold so quickly? Let's go through it step by step. The primary structure is the most basic level of folding and consists of a sequence of amino acids determined from the genetic code. The secondary structure describes the local structures that form due to interactions of the polypeptide backbone. Two major secondary structures emerge, alpha helices and beta pleated sheets, both of which are stabilized by hydrogen bonds. In alpha helices, every amino group bonds with a carbonyl group of an amino acid 4 units ahead, forming a helix. In beta pleated sheets, 2 or more strands form multiple hydrogen bonds with one another, forming a sheet-like structure. The next and final step is the tertiary structure, which describes the interactions of the amino acid side chains that form an overall 3-dimensional shape. Remember, side chains have many properties and can form bonds with one another. There are multiple interactions that determine the tertiary structure, but hydrophobic interactions are the predominant ones. Proteins fold in aqueous solutions, so in hydrophobic interactions, hydrophobic amino acids aggregate towards the core of the protein, where they're shielded by hydrophilic or water-loving amino acids. Tight packing allows weak van der Waals forces to form in the hydrophobic core, holding it together. After hydrophobic interactions, side chains in close proximity to one another form ionic bonds, covalent bonds called disulfide bridges, and hydrogen bonds. All of these reactions collectively help determine the protein structure. As proteins are able to fold quickly by focusing on local interactions first, then zooming out to focus on interactions occurring on a larger scale. For a long time, limited information of protein structures in our bodies has been a barrier in understanding how diseases like sleeping sickness work, because determining protein structure experimentally is a very time-consuming and expensive task. But thanks to AlphaFold, an artificial intelligence system, we can now predict protein structure given the amino acid sequence quickly and inexpensively. With this tool, we can expand our understanding of diseases. We can even design drugs that enhance or inhibit protein activity, which can help us cure diseases. And wouldn't it be awesome if we design proteins that break down waste like plastics or oils? There's a lot of potential here. But with a powerful tool in our hands, we'll be able to unlock the hidden secrets of these curious microscopic machines that make life work.